Hi everyone. In this video, we are going to learn about structures of nitrogen bases. We know that there are two types of nitrogen bases are present in DNA. So these nitrogen bases are called purines and pyrimidines. And when we look into the structure of these purines and pyrimidines, so these purines and pyrimidines are heterocyclic compounds. When we say heterocyclic compounds, these purines and pyrimidines are made up of two different types of atoms. So in case of these adenine, guanine, cytosine and thymine, so these two different types of atoms are nitrogen and carbon. Okay, so purines have two rings. So these two rings are made up of two different types of elements. They are called nitrogen and carbon. Total number of atoms are present in purines are 9, and total number of atoms present in pyrimidines are 6 atoms. So of these 9 atoms, 4 atoms are nitrogenous atoms are present, and 5 atoms are carbon atoms are present in case of the purines. So in case of pyrimidines, so 2 are nitrogen atoms and 4 are carbon atoms are present. Okay, so coming to this, the position of these atoms. So nitrogen atoms are present on the first, third, then also seventh and ninth position of purine rings, whereas these pyrimidines, where these nitrogen atoms are present on first and third position of the position of these particular bases. Okay, so the rest of the atoms, like in case of purines, the second one, the second atom, and also the fourth and fifth, then the eighth atom. So second atom, fourth atom, fifth atom, then also sixth atom. So these 5 are carbon atoms. So in case of the pyrimidines, so 2, 4, 5, 6, these 4 are the carbon atoms. Okay, so these are the purines and pyrimidines, which have the two rings and also the one ring respectively, with the two different types of atoms, and the position of these atoms also we see. So this nitrogen and the carbons, they are present at different positions in purines and also in the pyrimidines. So apart from this, what is most important to understand the structure of these nitrogen bases is their functional groups. So the functional groups are so important. So three types of functional groups are present in these nitrogen bases. The functional groups are amino group, and also we have the oxo group and methyl group. So see, three functional groups are there. So the arrangement or the position of these functional groups and these nitrogen bases is also so important. So in adenine, we see this amino group is present on the sixth carbon atom. So it is attached to the sixth, it is linked to the sixth carbon atom. So that is important. So this adenine has only one functional group that we have to understand. Okay, so the next two, such as guanine and cytosine, they have two functional groups. For example, you can see here, see it has oxo group and also the amino group. So two functional groups are present in the guanine and also the cytosine, where we can see the two functional groups are present. Okay, the numbering of these nitrogen bases is also so important. So we start giving number from the bottom. In case of this purines, so where we start from here. So this is a sorry, not from the bottom. So we start from here. So it is in a clockwise direction. This is the first atom, the second atom, third atom, fourth atom, fifth atom, and sixth atom. So the sixth carbon atom has the amino group. So this is the seventh atom, and the eighth atom, and ninth atom. So this ninth nitrogen atom, it forms a link with the sugar by glycosidic bond, by the glycosidic bond. So then this amino group and this nitrogen bases, so this amino group, functional group, and this this nitrogen, so these two, they form the hydrogen bonds with the adjacent base in a DNA strands. Okay, so we know that where bases, they pair, they form hydrogen bonds with the bases which are present on the opposite strand. So this amino group and also this nitrogen, so these two, they form the, they participates in formation of the hydrogen bond. Here this amino group, so it acts as a donor for the hydrogen bond. It is acts as a donor for the hydrogen bond, whereas this nitrogen, it acts as an acceptor for the donor bond that we discuss in the next video, what this donor and acceptor with respect to the hydrogen bonds. Okay, so in guanine also, there we can see here, here we have oxygen group. So this oxygen group, it participates in a formation of hydrogen bond, and also this amino group, it also participates in the formation of the hydrogen bond, and also this NH, that is that also participates in the formation of hydrogen. That's why the guanine forms 3 hydrogen bonds, and whereas we can see the adenine, it can forms the 2 hydrogen bonds only. Okay, the numbering here, it starts from the bottom, that in case of the pyrimidines, it starts on the bottom side. So this is the first atom, this is the second atom, third atom, fourth atom, fifth atom, and sixth atom. See, the second atom and the sixth atom has the bonds here. See, we can see this cytosine. So here, when we look into the cytosine, see here also this oxo group participates in the hydrogen bond, and also this nitrogen participates in the hydrogen bond, and this amino group also participates in the hydrogen bond. That's why this guanine and cytosine, they forms the 3 hydrogen bonds. So whereas in the thymine group, you know that, so adenine, which it can forms 2 hydrogen bonds. So here also we can see the thymine. So a second and this fourth, so these 2 atoms have the functional groups, and also the third one, which is a nitrogen molecule. So here what is most important is here that, so this oxygen and this functional group and this NH group, these 2 participates in the formation of the hydrogen bond. Okay, this is how they this is how these functional groups and the nitrogen atoms which are there, they participates in the formation of the hydrogen bond. Okay, so you know that, see, in adenine only 1 functional group is there. In guanine there are 2 functional groups, there are amino group and also oxo group is there. In cytosine also amino group and oxo group are there. In thymine, see, there are no amino groups, only it has oxo group. In addition to that, it has a methyl group on the 5th atom. So these 2 oxo groups, they are present on the 2nd and 4th atom of this heterocyclic ring. Okay, in uracil there, so these are present on the 2nd and 4th. Only this methyl group is absent. So that uracil, neither it has the amino group nor it has the methyl group. Only oxo groups are present. Okay, these are the structures of the nucleotides. Nucleotides bases. Okay, in the next class there we discuss about the structure of the ribose sugar in DNA. Thank you.